Okay, I think we're connected. Hopefully, people can hear me who've already joined. I'm going to wait another minute or so before I start the webinar, just to give anyone else who's going to connect a bit of time. And so we'll be starting in a couple of minutes, and then I'll turn my camera on. You'll be able to see me then. Okay, I think we'll get started. So welcome today to this webinar. I'll be presenting. I'm James Fels. I'm the head of applications development for ONI, and the subject of the webinar is all about immunology, specifically revealing immunology at the nanoscale using super resolution microscopy. And the aim of this webinar today is really to explain why super resolution can be informative in immunology. It's not so much a demonstration of ONI technology, although I will be talking about that towards the end. It's more a case of where within the biology of immunology are nanoscale events important? Why would someone want to interrogate them? And how have people done that in the past? And how might people do that in the future? So as I said, I'm the head of applications development. A little bit of background on my side. I'm an immunologist by training. I was a biochemist who saw the light and became an immunologist during my PhD, which I did with Professor Simon Davis here in Oxford, looking at T-cell activation. And I then continued to work with Professor Davis for a number of years, looking at activation of other lymphocytes and myeloid cells. And then I moved to Professor Mike Dustin's group, also in Oxford, again, looking at T-cell activation and active processes within that, using advanced biophysics and a lot of super resolution. And I've been at ONI for 2 years, where I've been heading up a lot of our biological applications development, much of which is focused on immunology. So I'm going to start with a bit of a broad overview of fluorescence microscopy. So apologies if this is a bit basic for some of you who might already be familiar with advanced imaging. But just to make sure that we're all kind of on the same page. Fluorescence is a term that most people are aware of, and most people have an understanding of how it might be used within immunology. It can be used for imaging, but also for flow cytometry, which has underpinned an awful lot of immunology, as we all know. It's essentially a way of using the unique photophysics of certain properties to enhance contrast. And the way that fluorescence works is that you have your fluorophore, which typically these can be fluorescent proteins, but they can also be organic fluorophores such as this, which tend to be the brightest, the most stable. And for the purposes we're going to talk about today, typically this is what would be used. Fluorophores, what distinguishes fluorophores from non-fluorescent molecules is that when fluorophores absorb light in one wavelength, they don't just re-emit it at the same wavelength. They actually emit a small amount of it as heat, which is this little gray arrow here, which means that when they re-emit the light, it's lost a small amount of energy. And because it's lost some energy, it means its wavelength will be longer, because shorter wavelengths of light have higher energy. And so what this means is if you shine a particular wavelength of light onto a sample, most of the molecules in that sample will re-emit the light at essentially the same wavelength, whereas just the molecules you're interested in will do it at a different wavelength. And if you can then discriminate those using a microscope and the relevant fluorescent filters, etc., it means you then have fantastic contrast where you literally only see the molecules that you're interested in. So this has been used in immunology an awful lot, as it has been in essentially all areas of biology. This has been used for standard resolution imaging, tissue imaging, where you can look at individual cells. You can also look at processes within cells. So again, you can look at, for example, the cytoskeleton, you can look at things moving around, and you can look at signaling events down to a reasonably good resolution of a few hundred nanometers, where you can see clusters of molecules and what they're doing. And this is extremely informative. As I say, fluorescence has also made a massive impact in immunology through flow cytometry. And there is imaging flow cytometry, which is making something of an impact in the field, but is limited a bit by the resolution, because it is very much the number of cells that you're imaging rather than the individual quality of each cell. So what's the problem? Well, the problem is this thing called the diffraction limit, which I'm sure, again, many of you might be familiar with. Just to contextualize this, if we consider a typical immunological process, such as the interaction between a T cell and an antigen-presenting cell, this is mediated by the interaction of the T cell receptor with peptide MHC molecules. And it's an exquisitely sensitive process. You only need a handful of peptide MHC molecules with the right peptide on it to activate your T cell and your TCR, and thereby your T cell. And when you look at this contact, this is called the immune synapse, it is highly organized. There are different molecules which are segregated into different parts of the synapse. This structure forms within about 5 to 10 minutes of the initial contact. And it is a fascinating process whereby you have T cell receptor typically in the center, which it forms microclusters, which then migrate towards the center of the contact. That's then surrounded by co-inhibitory or co-stimulatory molecules such as CD28, which is again flanked by adhesion molecules such as LFA1. And then we have inhibitory molecules or regulatory molecules, which are out towards the edge, such as CD45. And fluorescence imaging can tell you an awful lot about this structure. The understanding of the immune synapse has been instrumental in understanding how T cells get activated, as well as similar structures in other cell types. But the issue is that the actual molecular detail is happening down here on the nanometer scale. The TCR-MHC complex only has a lateral size of about 2 nanometers. And when you're talking about individual interaction events, which aren't biologically meaningful in immunology, the fact that you can't see these is problematic. So why can't we see them? Well, this is because of the diffraction limit. So if we take an image such as this, which is of a fairly typical T cell immune synapse, we can see the scale bar here, 5 microns. This tells us an awful lot about that interaction. However, when we're looking at this area, the real distribution of molecules may look something like this. There'll be a handful of molecules and they will be segregated from one another, or they might be interacting. We don't know. All we can see in the diffraction limited image is just a red area. And the reason for this, so on the left here, you see what the ground truth, and on the right is what you actually see. And the reason for this is the fault of this man, Ernst Aber, who described this process. This is because light, when it emerged from a point source, essentially spreads outwards, and it can diffract with other and interfere with other light sources that emerge from the same point. So this is very much the same problem that we have in cosmology, looking at distant things such as stars. Simply the light spreads out and blurs. And if two things are too close to one another, physically, it is impossible to distinguish the light coming from one or the other, because the light will interfere. And that is proportional to the wavelength of the light that you're using, but also something called the numerical aperture, which is essentially the range of angles over which you're able to capture the light. In cosmology, we get around that by just building larger and larger telescopes, because you essentially capture a wider range of angles, and therefore you improve your resolution, so you can get crisper images of stars and planets. But when it comes to microscopy, there is a technical limitation on how much you can actually increase your numerical aperture. And you can only work with certain wavelengths. You can go right down into very short wavelengths, for example, using electron microscopy. But for light-based fluorescence microscopy, you're typically working in the area of about 300 to 700 nanometers. And generally, the rule is that it's about half the wavelength is your resolution. So for a typical fluorophore, such as something in the 647 or the 488 channel, your resolution is going to be somewhere between 200 to 300 nanometers. So any 2 points that are closer than that, you will not be able to distinguish them. And as we pointed out in the last slide, the TCR peptide MHC is only 2 nanometers. So it's 100 times smaller than that resolution limit. So you're missing an awful lot of information. And this is just a physical constant of the universe because of the properties of electromagnetic radiation. There's nothing we can actually do to stop it. So the question is, how do we beat it? Well, as I say, we can't stop it, but there are tricks for beating it. And really, the realization comes that you don't actually need to beat it, or you don't need to get around it necessarily in order to know where your molecule is. And this is because if your molecule is this black spot in the middle, and then the spot of light that you're actually seeing is in the red, it's not just a solid spot of light that you see. It actually has a distribution which is reasonably Gaussian. So if you know for certain that you only have one molecule there, you don't need to say, oh, it could be anywhere within this 250 nanometer range. You can say with some confidence that it is within the central region. And you can apply statistics which will essentially localize this Gaussian distribution and give you boundaries which will tell you with most confidence this is where your molecule sits. And this is the principle of localization microscopy and other methods. However, the key thing, as I said earlier, is you need to be able to be confident that it is one molecule and one molecule only. Because if there are more than one molecule there, you're going to you're essentially going to localize them all to the same point, and they will look like one molecule, which may also not be in the correct position, because you will essentially end up with an average of the positions across multiple molecules. So the question there is, how can you be certain that one spot is one molecule? In the image I just showed you, there's no way to be certain that one spot is one molecule because it's essentially a carpeted fluorescence. So there's no way to distinguish one molecule from another. But there are tricks that will allow you to be confident that each spot you're looking at is one molecule, and thereby localize it with a very high precision that is down in the low nanometer range. So one way to do that is to simply separate your spots in space. So you can do that by having very sparse samples, such that the odds that any one spot overlaps with another is extremely low. And this is typically used for single molecule tracking or single particle tracking, where you tend to label a subset of the molecules that you're interested in and then track them over time. And you can then localize this. So you see on the right here, the blobs, you can see that's the actual fluorescence signal that you see, but then the traces are far more precise and fine than you would think from the blob, because at every spot in the video, the exact center of that spot is being mapped with high precision and then tracked over time. So this is a very effective way to see where these molecules are at very high resolution, in that you're observing individual molecules. However, the crux is you're only seeing some of your individual molecules. Most molecules that are active in immunology don't exist on this kind of level, on this kind of density. They're far more dense than this. So you are inherently going to deliberately ignore the vast majority of your molecules and then assume that the molecules you're looking at are representative of the larger population. And this has a lot of value when it comes to looking at single molecule dynamics and things like that. But it is limiting when it comes to trying to resolve complex structures, for example, where if you want to actually see the information, you can't just get rid of 99% of the molecules in the image. So how do we do that? Well, instead then, rather than separating spots in space, we separate them in time, which is an additional dimension that is open to us. But how do we separate spots in time? Well, if you imagine you have your sample, it has thousands, maybe even hundreds of thousands of fluorescent molecules in it. If you were to image them all at the same time, it would just be a complete carpet, and you wouldn't be able to distinguish one molecule from another. But in localization microscopy, we use various chemical and photophysical tricks to cause the molecules in the sample to blink, which means that in any one frame, only a very small subset of the molecules will be active. And in this video, obviously, this is coming very quickly, but in post-processing, you can take each frame of this video and localize each molecule. So as an example here, if we had a ring-like structure in the first frame, then we might have this molecule here at about the sort of 5 o'clock position turns on. In the next frame, it might be this one in 9 o'clock, this one in 12 o'clock. And if you capture enough of these until you've sampled the whole sample, you can then localize them all individually and build up a high-resolution image. So this is actually on the, what's blinking on the left is what you see on the right here. This is actin cytoskeleton in a COS7 cell. And you can see the blinking really doesn't give you much indication of what it is. But when you go and localize all the images, you can see this with extremely high resolution. And this is the fundamental principle for a range of techniques that are collectively called single molecule localization microscopy, SMLM. And this includes PALM, STORM, DSTORM, PAINT, and a few others. that you're probably familiar with. Today, I'm not really going to be going into the details of what differs between these different methods, but they're all founded on this same principle of localization. So what this means is that when with conventional microscopy, we can see down to about 250 nanometers resolution. With localization microscopy, we can see down below 20 nanometers resolution. And actually, in practice, it's typically quite a bit lower. A lot of the localizations that you get have down to 4 to 6 nanometer resolution. So it's an extremely powerful technique. And this is an example of the kind of greater insight that this can give you. This is actually an image of nuclear pore complexes. And obviously, you can see it in the conventional microscopy image. You can see that they're there. You can get a rough feel for their distribution, but there's literally no way to ascertain their structure. Whereas with localization microscopy, you can see their structure and you can get an approximate understanding of their size. So it is giving you an awful lot more information. However, it would be wrong to suggest that it's as simple as conventional microscopy. There are things you have to do that you don't have to do for conventional microscopy, which make it more difficult. So I guess the question here that we need to answer is why? Why would anybody do that? And specifically, why would anyone do that in the context of immunology? Well, this is why, because if you have greater resolution doesn't just give you pretty pictures, it gives you more information. If you have more information, then you can have a deeper understanding of the processes that you're looking at. And there are many processes in immunology that are founded on things that happen at the nanoscale. So we can look at them with a lower resolution and get a lot of insights, as I showed for the immune synapse with T cells. However, to really get to grips with what's fundamentally happening at a molecular level, we have to look at things at the nanoscale. And that's where super resolution comes in. So today in this webinar, I'm going to give you some examples of how this has been applied in immunology. It's worth saying at this point that a lot of the data I'm showing today was not collected by ONI or on ONI instruments. I will acknowledge the authors throughout. Most of this is published information from the literature. And it uses a variety of techniques, different super resolution techniques. I'm not going to go into the details of each one because more I want to get across the potential and the importance of super resolution as a concept. But obviously all of the references will be given, so please feel free to go away and read up the papers if you're interested. So firstly, one of the things that super resolution can do in immunology is to reveal subcellular structures. As you can imagine, most subcellular structures have some level of contribution at the nanoscale, and they may be dynamic or they may be highly organized at the nanoscale. And so understanding them through super resolution can be very informative. So as an example here, this is a case of the degranulation process that occurs in mast cells, and this was done by Marco Fritz group. And this was actually taken using a form of super resolution called structured illumination microscopy. Here, they've been looking at the dynamic processes that underpin actin reorganization within mast cells as degranulation occurs. And in cyan, you can see the actin, and then in magenta, you can see an Annexin-5, which is essentially a marker for the fusion of intracellular vesicles with the surface. So this is a degranulation event. And you can see here that at the beginning of this process, at T0, you have a very dense actin network, which is fairly typical for most cells, most lymphoid and myeloid cells. However, when the activation occurs, this is occurring on a flat surface, the actin very quickly reorganizes and essentially moves out of the way to allow efficient directional degranulation in the direction of the activation source. And one of the things that is possible through this is a dynamic assessment. So this is actually what happens when you see it live or as a video. You can see the timer at the top there. This is actually in seconds, not minutes. They are seconds. So this, you see again here, the very dense actin network restructures. And not only does it move out of the center, but you can see at the periphery, you get these new structures forming, which are very dense here. They're very dense and they have a very different actin organization to the center. So there are multiple different actin species that occur here. And they're only really possible to see these through super resolution microscopy. So we can see these kinds of subcellular structures, but we can also look at subcellular structures that potentially report dysfunction. So in this case, we're looking at cells that have been infected with Ebola virus or not infected with Ebola virus. And this was done in collaboration with Professor Becker in Marburg. And what you can see is there's very clear distinctions in the actin cytoskeleton within healthy and infected cells as a consequence of the infection. Now, actin, it's possible you'd see some of this through conventional resolution, but you wouldn't be able to see the fine detail of what's going on in these infected cells. However, actin is actually quite a clear tool. There are many, many processes where dysfunction would in no way be visible through conventional microscopy and would only be visible through super resolution methods. The other thing that we can see here is we can correlate the processes that lead to this dysfunction. So even though we may be able to see some of the outcomes of dysfunction using conventional microscopy, it would be very hard to see the mechanistic causes of why you have this dysfunction. And so here we can see, we can correlate the different points of assembly of Ebola with the actin cytoskeleton. And thereby over time, we can build up an understanding of how the assembly and distribution of these viruses within the cell leads to changes in the cytoskeleton, and thereby understand the mechanisms behind viral infection in these cells. So there are many other examples of subcellular structures that can be informed in immunology through super resolution microscopy. But I would like to go next on to receptor clustering. Now, receptor clustering is really almost the ideal model for using localization microscopy, because inherently you see one molecule at a time and then you build up your image. It's absolutely perfect for looking at the nanoscale clustering behavior of various molecules. And one of the hallmarks of immunology that's becoming increasingly evident over the last decade or so is that immunology is in many parts a story of what happens at the cell surface. The immune system is extremely complex, as we all know. It's largely driven by cell-cell interactions, and those cells typically communicate either through soluble factors or direct cell-cell interactions, which all take place at the cell surface. And the clustering of various molecules is extremely important in regulating them and defining what's going to be their downstream outcomes. So as an example, we can see here that amplification is often dependent on receptor clustering. Anyone who's worked in signaling of surface receptors in immunology will understand this point, that it's very, very common that you have a receptor, could be an antigen receptor, a chemokine receptor, or something else like this. Often the model is that the activation of that receptor leads to some kind of post-translational modification, typically a phosphorylation event, which then leads to the recruitment of effector molecules and then adapter molecules and then effector molecules. And typically the cell has a threshold for activation below which it will not respond, but above which it then triggers some marked change in the cell. So for example, in the case of T cells, this would be the T cell receptor. If it's sub-threshold signaling, the cell as a whole does not activate, but then once you reach a level of stimulation of the TCR, the whole cell becomes active and launches a full T cell immune response. And often what is the determining factor in this is the degree of clustering of these molecules. And this is because the effectors and the adapters often contribute to post-translational modification. They're commonly kinases, and therefore having them in a very dense area causes cooperative amplification of the initial signal. And so you may have a distribution of molecules which at the top, if it's slightly more sparse, is not signaling. But if it is just that little bit sparser, enough to tip across the threshold, it is suddenly a signaling cluster. And the difficulty is that this occurs on the nanoscale level. So the size across these two clusters that I've drawn could easily be 50 to 100 nanometers. So they would look exactly the same if you were viewing them through conventional imaging. It's only through super resolution microscopy, and particularly through localization microscopy, that you can really see the fine detail here and thereby understand how these things come together. Similarly, inhibition is extremely dependent on what happens at the nanoscale. Generally, inhibition in immunology, if we're talking about surface receptors, is driven by co-inhibitory receptors such as PD-L1, CTLA4, these kinds of things. And they generally work by the recruitment of soluble inhibitory phosphatases, such as SHP1, SHP2, and other inhibitory molecules. And essentially, they are then limited by the molecular reach of the tails, the cytoplasmic tails of those molecules. If you have a PD-1 molecule and it is going to inhibit a CD28 molecule, it can only inhibit those molecules that are within the dynamic reach of its cytoplasmic tail. Because if it is outside of that, then any SHP2 that it recruits will not be able to bind and dephosphorylate the CD28. And this was shown by a very nice study by Omar Dushek's group, where they characterized how the molecular reach of different inhibitory molecules impacts their ability to inhibit local molecules. So you can clearly see then that even if you have the same distribution of CD28, which I'm representing here in yellow, different distributions of PD-1 will have different outcomes. In this case, a very clustered PD-1 will essentially have a lot of redundancy. A lot of the molecules of PD-1 will be very close together. Therefore, they won't really be able to add anything when it comes to an inhibition. They'll all just be accessing the same CD28 molecules, so the degree of inhibition will be comparable. Whereas the same number spread out more sparsely among the CD28s will essentially cover a broader area of the cell surface and therefore lead to different levels of inhibition. Similarly, you could have very structured organization of these inhibitory molecules leading to a structured inhibition, which might be important, for example, in polarization or directional behavior of the cell. And this is not conjecture, this is known to happen. So, for example, here in a study from Takashi Saito's group, we can see how PD-1 is organized in T cells as they get activated. You can see here on the right, on the left, you can just see the cell, the total cell contact, and on the right here, you can see the distribution of PD-1. If I play it again, you can see this is very dynamic. The PD-1 clusters and then goes into very organized domains. So you can imagine that at different points in this process and at different areas of the cell, the degree of accessibility of PD-1 to things that it is going to inhibit changes, and therefore the inhibition changes. But you can only really see this at the fine scale using super resolution imaging. This image is extremely informative for the global distribution of PD-1, but it won't do anything to tell you about individual cases of PD-1 inhibiting CD28. And this is extremely important across many, many molecules in immunology. As I'm sure many people here are aware, surface receptors in immunology are very modular, and many of them are built around this ITAM-ITIM system, or sometimes ITSM system, where you have specific phosphorylation sites, tyrosine phosphorylation sites, which are then anchors for a more limited range of effector molecules, such as kinases and phosphatases, typically. And these are used across many, many molecules. And so the same principles of clustering and molecular reach apply to all of them. They are also applicable to engineered molecules, such as chimeric antigen receptors, which tend to be engineered on a fairly modular basis by chopping and changing different signaling domains from various natural receptors in order to elicit a particular response. So, for example, where the docking site for a ZAP70 molecule or a SHP1 molecule is on the length of a particular chimeric antigen receptor will impact how it clusters. It will impact how it has molecular reach on the surrounding molecules. But again, all of that is only accessible through super resolution microscopy. So as an example, here we can see a study from Dan Davis's group where they were looking at macrophage activation, looking at FC receptors, which are one of the many molecules that have ITAM-ITIM mechanisms. And this is a FC gamma receptor 1, so against IgG. What we can see here is that the inhibitory molecule SERP alpha, this was studied using D-stone microscopy, in an unstimulated cell is very closely co-localized with these small clusters of FC receptor. And because SERP alpha is inhibitory, this means that the activity of the FC receptors is kept low. However, upon activation, we can see that the inhibitory regulator segregates from the FC receptor. And this is informative about the mechanism by which these FC receptors become activated. And again, this kind of information, which is occurring here, you can see in this plot on the right, the distance. We're talking easily below the diffraction limit. so only accessible through super resolution. Similarly, the clustering of different molecules can vary substantially between cell types or between conditions. And so really understanding this is quite important when it comes to understanding how different things are likely to behave, both in vitro and in vivo. As I mentioned earlier, clustering of molecules will cause potentially different outcomes. So the degree or the nature of the clustering is extremely important. And that doesn't just mean the molecule itself, but it can mean, of course, its ligand. If the ligand that a particular receptor is binding to shows different levels or different types of clustering behavior, then that will induce different types of clustering behavior in the receptor, which will then potentially cause differences in its downstream behavior. So in this study, which again was from Dan Davis, they were looking at class 1 HLA on different cell types, natural killer cells, T cells, and B cells. And they observed that the density and clustering of these molecules differs according to the cell type, even for the same class 1 molecule, which means that these different molecules may induce different sensitivities for cognate TCRs on T cells that are interacting with these cells. Because you could imagine, for example, a cell that has very highly clustered class 1 MHC might have a lower threshold for activation of the corresponding T cell than one where it's extremely sparse. Because when the binding occurs, you are going to induce very robust clustering of the TCR even before you've activated the T cell. And this isn't just cell type differences, but this can also cause genetic differences. So again, from Dan Davis's group here, looking at a storm of different allotypes of KIR molecules, we can see that the different densities and different clustering behavior correlates with their genetics. So different allotypes of these molecules causing different clustering, which again can lead to differences in their downstream behavior. So this helps to inform our understanding of the subtleties of immunology and how seemingly similar things can have very, very different outcomes. It's not uncommon in immunology to have things that look exactly the same insofar as our understanding, but then they have very different outcomes potentially if you put them into a patient, for example, if it's an engineered receptor. And it could be something to do with their nanoscale organization that just has not been characterized. So this is another example here where we have, this is data from ONI, where we have an activated B cell stained for CD20. I put this in to give an indication of the kind of information that you can expect to get through clustering. So you can look at things such as the cluster size. You can look at their various locations within the cell. You can also look at the density, so the number of localizations within a cluster, but also the number of clusters within a given region. And not shown in this image, you can look at co-localization between different molecules. You can do this in tissue as well. So here we've got tumor profiling. I can't go into the details of what exactly is being stained here, but these are different targets for chimeric antigen receptors, and these are different patients. One of the clear things here you can see between the two patients versus healthy tissue is that there's a lot more of the CAR target. So that in itself is informative, but arguably is accessible through more conventional IHC methods. However, you can also see the degree of clustering and the style of clustering across different patients. And it's perfectly possible that you might have two patients with exactly the same total amount of a given target. However, the clustering behavior may be different in one versus another, which may then heavily impact the potential outcome of any chimeric antigen receptor therapy or other immunomodulatory therapy that may be applied. And this is an area of patient characterization that currently is not really accessed outside of academic studies. Here's another example of slightly more complex tumor profiling, because it is possible, of course, to correlate the different structures that you see with the ultrastructure of the tumor itself. So in this case, this is just laminin and mitochondria within a tumor section from colorectal tissue. And you can see at the nanoscale how these different structures organize. So it may not be enough to simply say a molecule is clustered or not clustered. It might be, is it clustered in a particular area? How far is it from something else? All of these things are extremely difficult to see. It takes a lot of effort. You have to do super resolution and see it. However, the fundamental reality is these are the things that determine the immunological outcome. So we ignore them at our peril. And there's been increasing interest in recent years at tumor-infiltrating lymphocytes and how these kind of things impact particularly cancer responses. The field of immune checkpoint inhibition is obviously very large when it comes to immunotherapies. And you can see here that there are many co-stimulatory and co-inhibitory receptors that are present on tumor-infiltrating lymphocytes. Here's just a summary on the left, and this on the right is a study from Tao Dong's group looking at the different positivity fractions for different tumor-infiltrating lymphocytes in blood and in tumor tissues. The takeaway is that these molecules, which all function through ITAM-ITIM mechanisms and therefore are very subject to impact from clustering, are extremely important in the anti-tumor response for, in many cases, both solid and liquid tumors. Similarly, when it comes to receptor clustering, we don't have to limit ourselves to the cells themselves. There's been increasing interest in recent years in extracellular vesicles and their role in immunology. This study here from Wei Go's group back in 2018 showed that a large part of the immunosuppressive response in various tumors is due to PD-L1 on exosomes, not on the cell itself. And this is in part because the exosomal PD-1 is in effect clustered because it is on an exosome. It can be released. And here on the right, this is from a review from Fangfang Zhu's group. You can see that this has many modulatory functions on the immune system. And so understanding how these molecules are incorporated into EVs and how they are presented on EVs is, again, important for understanding how immunology, particularly immunosuppression by cancers, is impacted. And the problem with exosomes and EVs in general is that they're very small. They are well below the diffraction limit. They're typically about 40 to 150 nanometers in size. And so if you look at them through conventional imaging, you will just see a blob. However, when you're using super resolution microscopy, again, using DStorm as an example here, you can start to see many features that are important. So this is data taken in-house from ONI. These are from colorectal cancer. There's nothing particularly immunological in this exact example, but hopefully you can get an appreciation for the sizing that you can do using super resolution here, and also an understanding of the abundance and potentially the location on the EV of different molecules, which in this case are CD63 and CD81. And in this case, wheat germ agglutinin was used to stain the glycocalyx of the EV so that you can more easily see the outline. So here we can then plot, for example, the size of the EVs, how that changes, but also the relative abundance of different molecules on it. So that could be PD-L1, that could be diagnostic in the case of direct patient care, or it could be for research purposes in terms of understanding how the immune response changes with different treatments. Similarly, I want to talk a bit about dissecting complexes. We could argue that EVs are complexes as such, but generally they're considered more sort of particles, other bodies rather than a complex. But there are lots of particulate complexes which are purely protein-based, which are becoming more and more interesting to immunology. It's only been a couple of years since the discovery of supramolecular attack particles, which are released by T cells and other lymphocytes. And these are essentially dense particles of protein that get released by killer T cells onto their target. And they contain most of the cytotoxic elements that are used to kill infected cells. And they're sort of like time-release grenades where rather than the cell releasing lots of very cytotoxic soluble factors, it releases them as a very dense particle, which is safer for the cell and surrounding uninfected cells. And in Mike Dustin's group that originally characterized this, they used STORM to characterize the structure of these particles. So here we can see how these particles contain perforin, which is extremely cytotoxic, and granzyme B, and then has this architecture of thrombosbondin, which helps to hold the particle together. And so this was a very impactful paper. It was a novel mechanism of cell killing that had not previously been understood, and it was only able to be reported because of super resolution. This has gone on, and Dan Davis's group have reported the same particles in natural killer cells, And so it is quite likely that similar particles exist in other lymphocytes. And actually in Dan's group, they were able to correlate different types of stimulation of the cells with different properties of these particles, both in terms of the number of particles that were released, their size, and their cytotoxic molecule content. Again, all of this only accessible because of super resolution imaging. Similarly, when it comes to dissecting complexes, we can look at quite complex complexes such as viral assembly, and it's possible to look at the co-localization of different viral molecules, how they come together. Again, this was a collaboration between ONI and the University of Marburg, and we can look at different sites of virus assembly, these viral factories, and potentially look at where different levels of maturity of the virus exist within the cell, which again can heavily inform the biology of the virus and potentially contribute to antiviral development. There are also very dynamic signaling assemblies. So these are complexes which come together very quickly and then break apart. This is from a study back in 2013 from Lawrence Samuelson's group. Ironically, my resolution of the figure is not good, but the resolution of the experiment was excellent. And what they've looked at here is the different stages of cluster formation, signaling cluster formation in T cells as they activate, and the recruitment, firstly, the phosphorylation of different molecules, and then the recruitment of different effector molecules over time. And they were able to map out this process where we go from an unactivated cell to initial contact, which leads to phosphorylation of certain molecules, recruitment of effectors such as ZAP70, first order clustering, and then later order clustering, where you end up with an end cluster, a mature cluster, which is very different from the original clusters and has quite distinct clustering and signaling behavior. And this information is out there at the moment for a number of natural receptors, such as in this case, the T cell receptor. However, for engineered receptors, this generally doesn't exist and probably is very different from between each receptor and the next. And understanding the series of events that occur in the recruitment of signaling effectors to these various receptors, over what timescales, in what order, spatially, what's happening, is really important, again, to be able to understand why some receptors work well and others don't, because without that information, we're essentially ignoring a fundamental part of their behavior. And similarly, lastly, we can look at subunit organization. So we might have complex clusters of molecules which come together stably, and we can look at their organization both in terms of the relative numbers, so the stoichiometry of different molecules, and potentially their spatial distribution. So in the interest of time, I want to just quickly move on to why do we not do that. Hopefully, I've convinced you that this is an important these questions are important. They really matter for fundamentally understanding immunology, and the techniques that are out there can answer them. So the question is, why don't more people do this? Because it is still reasonably niche. And the short answer is it's quite hard. There are a lot of difficulties with super resolution. The first one is the equipment. The equipment for super resolution on the whole is very complicated. It is often homemade. Even if it is commercial, it often requires lots of maintenance. It requires technical experts to use, operate, maintain. And just as an example, this was a super resolution instrument that I used to work with. It's an excellent home-built system, really good. I got a lot of very good data out of it, but it is nonetheless complex. So you have to have a separate computer, there's a camera, you have standard components such as the lens, eyepiece. It requires a table for stabilization so that as you can imagine, if it moves at all, that's going to completely ruin your image. The optics are often exposed, the lasers are often separate. And usually this needs its own room because it has to have a dark room. And then when you go to do your imaging, you turn off all the lights and make sure no one opens the door. The other issue is expertise. Sample preparation is not quite as simple for super resolution as it is for conventional imaging, which already has some complexity to it. So people need to understand what they're doing if they're going to be able to design and follow up on an experiment. It's technically difficult to use the equipment, as I've said. The things like understanding the buffer requirements and preparation is also challenging. And simply knowing what good images look like. You have to be able to identify good versus bad data. Otherwise, you're at risk of presenting bad data. Similarly, with the analysis, you have to be able to reconstruct the images from the raw data, and you have to be able to extract the quantifiable data that you want from the image. Again, all of this requires a lot of expertise. So these are the general barriers. I want to quickly talk about how ONI is aiming to bring down these barriers in many ways. So we essentially exist to bring the information of super resolution to as many people as possible. And what we mean by that is not bringing a microscope to as many people as possible. It is the information. We're not just an instrument provider. We are a provider of information and solutions. Nonetheless, a key part of that is our equipment. So in our case, we have this super resolution compatible microscope, which is the Nano Imager. It does not come with all of the difficulties that I just described. It is very small. It can fit inside a biosafety cabinet if you want to do something that is potentially a biohazard. But this is the size here you can see on the left. It fits onto any conventional benchtop. It does not require its own darkroom or stabilization table or anything like that. And it is, despite being extremely high-powered and actually more high-powered than many of the competitors on the market, it is very straightforward to use. And so it doesn't come with many of the difficulties of use that I already described. Similarly, we need to not only provide instruments, we need to make it so that everyone can understand how to use our instruments. So we have resources such as our university. We have guides to super resolution imaging that allow people to go on to understand how they should be designing and doing their experiments. And we have a range of resources that are available to people. And we also have a lot of in-house experts who are able to advise and help on the design of experiments. So we're very much not the kind of company that just sells an instrument and then says bye to our customers. We like to be involved with our customers throughout the whole process and supporting them as much as we can. Similarly, we directly help people through sample preparation support. We have a number of kits which are specifically designed to enable easy sample preparation towards a particular question. We have these for extracellular vesicles, which I mentioned earlier, but the one that I'm showing here is actually for lymphocyte sample preparation kit. So this is just an example of the kind of information that someone could get if they were using this. But this is essentially a kit that allows the capture, the fixation, and the probing of a range of different lymphocytes in a way that is biologically meaningful, does not interfere with the cells, doesn't activate them, etc., and is optimized to give very, very high quality samples for super resolution imaging. And we have, as I say, we have a number of kits that are out there for different biological questions. And we very much see it as part of our mission to enable the use of our instruments through these kind of kits and consumables products. Similarly, we aim to assist with the analysis. We have streamlined data processing. This is actually the software that runs on the instrument, but we also have other software packages which are specifically designed to answer questions across different areas of biology, and to do so in a way that does not require much in the way of understanding, simply an ability to identify which tools would be the best to use. And again, we are there to assist with that. So that's our mission. This is where we see super resolution imaging going in the future. In the past, we had this kind of level of information where back in the day, you could just about see what was there, but it was hard to see much detail. And then over time, we got more information. And then going forwards into the future, we'll be able to see exquisite detail. And I started this talk by saying that microscopy is an awful lot like cosmology. And so I'm ending on an example for Pluto, which back in 1996 was just a blob. And now we can see all of this lovely detail. It's exactly the same when it comes to microscopy. And more fields are moving to this lovely crisp end of the spectrum. And that includes immunology. And hopefully I've done a bit today to convince you of the need for that. And hopefully you'll be able to consider using some of these techniques in your own research. So I'm going to say thank you at this point, and I think we are going to swap over to any questions that might be had. So I'll just come out. Let me see. So we have a question here, which is, can you use lipid dyes for the imaging of extracellular vesicles? You can. It is somewhat challenging, and there are considerations that have to be had. One of the things is that generally lipid dyes form micelles, which are about the same size as the EV. So that often these micelles are 20 to 30 nanometers in diameter. And that causes two problems. One is it means that the rate of incorporation is often not good. Generally, when you use lipid dyes to stain a cell, it's not free dye molecules inserting into the plasma membrane of the cell. It is a fusion event between the micelle and the cell itself. So there aren't many free molecules floating around, which means it's quite challenging to get the EV and the micelle to fuse, because you can imagine the sort of curvature is a block. The other thing it means is you add an awful lot of molecules to your EV, and there are several studies out there that show when you use lipid dyes, you actually just increase the size of your EV because you put so much additional material into the EV. So it is possible, but it's not straightforward. And generally, we try and avoid that, and we use more methods that do not directly impact the membrane. We have a question here. Chips are rather expensive. Can it be done without the chip? Our chips aren't that expensive, but in principle, yes. However, I would say that if specifically this is to do with lymphocyte imaging, arguably the biggest challenge in imaging lymphocytes is actually the capture method. The problem with many lymphocytes is that essentially you put them on any hard surface, they start to activate. And this is due to the size-dependent segregation of molecules at the cell surface. So, for example, CD45 is a very large phosphatase. As soon as the cell starts to come into contact with a glass surface, for example, if it's been coated by polyol lysine or something like that, the CD45 gets pushed out simply because it's large. And because it's a key regulator of molecules on the cell surface, that then changes their behavior. And then the question here, what about EVs? Sorry, so maybe that was in the context of EVs. Again, it's a difficult challenge with EVs is how you capture them. So in principle, yeah, you could use our resources without the capture molecules. So if you have a particular capture method that you prefer, then that would be fine. You could use our material. However, we've generally tried to, for these kits, come up with an integrated solution where all the components work together really well. And we've tried to optimize around a lot of the problems when it comes to capture. So we have multiple different capture methods that are available for EVs. So it's possible they would overlap with the kind of custom capture that people are doing anyway, in which case it could just be swapped out. But yeah, as I say, any of our components is in principle available on its own. So there's a question here, which is, I'll quickly answer this question about the subpopulation, because that's related to the last one, and then I'll come back to the vibration table. So the question is, does the kit miss any subpopulation of EVs? The short answer to that is not really. We generally capture in a way that's pretty agnostic to the biomarker composition of the EVs. However, it is in principle possible that depending on the sample, there could be subpopulations that have never been encountered and would not be captured as efficiently as others. However, because we offer a range of capture methods, what we would generally suggest is that people try a couple of different ones in order to try and tease out any particular subpopulations, such that if it's missed by one capture method, it'll be picked up by another. But generally, when we've designed the capture method, we've tried to be as broad as possible so that there'll be real minimization of loss of different subpopulations. And then there's a question here about does the nanoimager require an anti-vibration table? No, it doesn't. So it's very stable in and of itself. You can put it wherever you want. People have actually imaged in the back of vans in the past, which, as you can imagine, is slightly shaky. We at one point were exploring putting it in a medevac helicopter so that you could use it for diagnostics of people being evacuated from war zones in a helicopter. So I think that says a lot about how stable the system is. And then the other question is, does it support multi-well cover slips? It will support multi-well cover slips, provided that they are not, it wouldn't hold, for example, a 96-well plate, but you can get these multi-well cover slips that are sort of the size of a standard BD, and that will fit fine onto the stage. The objective has a 2 centimeter by 2 centimeter range of movement. So anything where you're imaging wells that are kind of on that sort of spatial scale would be absolutely fine. But actually the imaging range is slightly larger than that because you can, of course, move your chip as well as moving the stage. So typically anything that would fit on normal BD or slightly larger than that would be absolutely fine. If there's any more questions, feel free to pop them in the chat and I'll do my best to answer them. So the question is, what's the lowest resolution that we can resolve? So in principle, there is no lowest resolution because the resolution is when it comes to localization microscopy is kind of on a molecule by molecule basis. So we tend to talk about precision when it comes to localization microscopy. The more light you get from a particular molecule, the more stable it is, i.e. is it moving much in your sample, what's the background, etc. All of that contributes to the signal to noise. So something with an extremely good signal to noise can easily have a very, very clear Gaussian distribution, which means you can localize it with a lot of precision. And it's not uncommon in our images that you're getting localizations down to about 2 nanometers. However, more typically, as I say, it's generally between kind of 4 to 15 nanometers resolution is probably the majority. And then we generally set a cutoff at 20 nanometers. So anything that localizes poorer than 20 nanometers, we then remove from the data because it's not good enough. However, the software allows you to set your own parameters. So you could easily remove, be more stringent or less stringent according to the need of the experiment. I'm glad people find this informative. Thank you. The question was, what's the function of the storm buffer? So the storm buffer, in this case, talking about D-storm, you need the buffer in order to induce the blinking behavior of the dyes. Most of these dyes don't blink spontaneously if you were to image them in PBS. Some of them do, but most of them don't. They would just be continually active until they bleach. But the storm buffers have within them a dark state stabilizing agent, which essentially pushes the molecules from being fluorescent to dark, but not bleached. Because the transition from fluorescent to dark is reversible, so it will come on and off and therefore blink, whereas fluorescent to bleached is irreversible, so it's off forever. So it stabilizes that, and therefore you get blinking. It also adds to the photostability of the molecules, and so you can image much longer without them bleaching. We have a question here: how many colors can you look at simultaneously? Well, the system has 4 different lasers. So in principle, all 4 of them can be on simultaneously. And it captures 2 halves of the camera at the same time. So generally, we recommend imaging 2 colors simultaneously, but in principle, you can do more. When it comes to D-storm imaging, we generally image 1 color at a time, but that's because usually you're looking at fixed samples, so it's not really advantageous to do simultaneous imaging. But for live imaging, that's perfectly possible. And then there's a question here: can it do live cell imaging with drift correction? So yes, so it can do live cell imaging, and drift correction is inbuilt into the software, so that would be done post-processing. Drift correction is most effective on fixed samples because the drift correction algorithm looks at points over time and then tries to track whether they've moved in various dimensions over time. However, if you were doing live cell imaging, it would be a bit more complicated because things would be moving. But if you're looking at a large structure like a cell, which is what you would be doing with live cell, then the drift correction would work. However, compared to other systems, we don't actually need very much drift correction because it's an extremely stable system and there's very minimal drift. Particularly if you were doing something like live cell imaging, the main thing would just be to let the system equilibrate to 37. And once it's done that, the drift is very minimal. And actually for live imaging, the sort of spatial scale of any potential drift will probably be well below what you're actually interested in. So there was a question here: what's the molecular process for which, in your opinion, you can get more advantage from super resolution in immune cells? Well, for me, it's probably the clustering and the looking at the co-localization of different molecules. So as I discussed, this kind of signaling behavior, whether you, for example, on a car, do you get predominantly ZAP70 recruited, or do you get PKC alpha? You know, these different molecules. We don't know this for most of these things, and for sometimes people put a domain into a car and it makes no difference, and it's possible it's just not recruiting the thing it's meant to recruit. So I think that is really very high potential, because as I mentioned, for localization microscopy, you see one molecule at a time. So you really build up the cluster and you can see these details in really effectively. And the follow-up to that was, have ever looked at neutrophils? I have, a long time ago, not at ONI, but neutrophils, again, well, they're fascinating cells anyway, but they have all of these kind of these receptors that I already talked about, like FC receptors, complement receptors, that are subject to clustering. But it would be very interesting to look at dynamic processes in neutrophils, because obviously they degranulate like crazy and turn themselves inside out and all sorts of things. I imagine there's an awful lot of nanoscale organization there that would be really interesting to look at. And then question here, I didn't talk much about Palm and how do you achieve Palm with the nanoimager. So a quick recap for people who aren't familiar with Palm. Palm is another super localization-based method, but instead of using spontaneous blinking like you get for DStorm, Palm uses deliberate photoactivation of molecules using an extra light source, which is generally the UV light source. So you can do Palm on the nanoimager very simply because it has a UV laser. All you do is you have your photoactivatable molecules. They are generally all off at the start of your image. You then turn on the UV. You can either do it as pulses or you can have it on continuously at a very, very low laser power, which means that they will stochastically activate. And then you acquire the data, you acquire the information with your excitation laser on as well, which would be one of the, like the red laser or the yellow laser. And then they effectively blink. What's actually happening is they're not blinking. They are getting activated stochastically. However, it amounts to pretty much the same thing. And then all the data analysis is exactly the same. So Palm is completely possible. And then we have here, how can we resolve if two colors are almost the same range? Well, if you have two molecules that essentially activate to the same extent with the same laser and have very, very similar emission spectra, then you can't really resolve these. We are working on spectral segregation of different molecules that have very similar excitation and emission spectra. But at the moment, if they are very similar, you can't resolve them. The main consideration is how are they excited, because if they have the same emission spectra but are excited quite differently by different lasers, then you can segregate them quite easily by just writing or constructing the experiment in such a way that the activation lasers are on at different times, and therefore you can discriminate them quite easily. I see that we're essentially at time. I've maybe got time for one last question if there are any. And I'm just going to quickly check that I haven't missed anyone's questions. Okay, great. Well, thanks everybody for coming. Please check out our website. If you would like to get in touch with us about anything, please go to oni.bio/contact, and you can reach out to us for sales information or scientific advice or anything else you want to discuss. We're here to help. And if anyone wants to talk immunology, I'm always keen to do that. But otherwise, I will just end it there and thank you for joining. The recording of this will be available. So if you do know any colleagues who you think this would be informative to, please point them in our direction. So I'll say thank you there and wish you all a very good day.